The third step in aerobic respiration, so remember the first step was glycolysis, and the second step was the oxidation of pyruvate. So now the third main step is the citric acid cycle, which used to be called the Krebs cycle. The citric acid cycle occurs in the matrix of the mitochondria, so it occurs in the same place as the oxidation of pyruvate. So this is that kind of gooey filled center of the mitochondrion. Now for each glucose molecule that goes through glycolysis, the citric acid cycle is going to occur 2 times. And that's because, remember, we started out with this 6-carbon glucose that then was kind of split in 2 to form 2 3-carbon pyruvates. And then those 3-carbon pyruvates got converted to 2 2-carbon acetyl-CoAs. So taking a look at the overall reactants and products of the citric acid cycle, we're going to start with our 2-carbon acetyl-CoA that goes in. Now that 2-carbon acetyl-CoA goes into the citric acid cycle, and what happens is it ends up releasing that coenzyme A, and one of those carbons breaks off to form carbon dioxide. So we lose another carbon here in the form of carbon dioxide. Now that acetyl-CoA combines with a molecule in order to form another molecule called citrate. And citrate is also known as citric acid, which is how we get the name citric acid cycle, because the first compound produced in the cycle is citric acid. In addition to kind of rearranging and juggling some carbons, we also make some ATP in the mix. So you can see that here that we make some ATP. Now this ATP is made similar to the ATP that was made in glycolysis. So this is also known as substrate level phosphorylation. Additionally, we made some NADH. So remember, NADH is our electron carrier. So we're going to add our little dots to remind ourselves that there are electrons there. So remember that when NAD plus is reduced, reduction is gain. So it's gaining electrons in order to form NADH. Now, in addition to NAD plus, there's another electron carrier called FAD. And it works the same way, where FAD is reduced, which means that it picks up electrons, so it gains electrons. So it's reduced, and reduction is gaining electrons. So once again, we can treat it very similar to the NADH. It's just a molecule, but it's a different molecule than NADH, but the outcome is the same. It picks up some electrons as these chemical reactions are occurring, and it's going to end up carrying those electrons to the electron transport chain. So if we look at the products of the citric acid cycle, let's take a look at what happens to these products. So first of all, CoA, remember this is coenzyme A. And coenzyme A is needed for the oxidation of pyruvate reaction. So it actually gets recycled and it goes back to be able to use to oxidize pyruvate again. The carbon dioxide that was produced gets exhaled. So once again, it first goes into the bloodstream, and then as carbon dioxide builds up in the bloodstream, some of it gets exhaled to get it out of the body. The ATP that we made is used by the cell for energy. So we made a little bit of ATP during glycolysis, we made a little bit during the citric acid cycle, and this is ATP that's ready to use by the cell. We also made the NADH and the FADH2. Remember that both of these picked up electrons, and they are known as electron carriers, and they're going to carry or transport those electrons to the electron transport chain. And what's ultimately going to happen is that transferring these electrons to the electron transport chain is ultimately going to allow these molecules to help the cell make a lot more ATP. So now we're moving on to kind of the fourth major step of aerobic respiration. And the fourth step, kind of altogether, we call it oxidative phosphorylation. But remember that oxidative phosphorylation actually kind of occurs in two separate steps. And so the first step is the electron transport chain. And then the second step, I'll go ahead and put it here, is called chemiosmosis. And we'll talk about that a little bit later. So starting by looking at the electron transport chain, we'll take a look at where it's located. So the electron transport chain is actually made up of these transmembrane proteins, which we also learned are called integral proteins. All right, so this chain is just a chain of proteins. So there's multiple proteins that are kind of lined up. And these proteins are embedded in the inner membrane of the mitochondria. And remember, we said that this inner membrane is called the cristae. And the cristae are these folds that go around here. And remember that this inner membrane is folded in order to increase surface area. Because if we increase surface area, we have more room for electron transport chains. And the more room we have for electron transport chains, the more ATP we can make at the same time. So it really helps to make ATP synthesis more efficient. And so these electron transport chains are these kind of proteins that are basically embedded in this kind of inner membrane here. All right, so each set of these would be kind of representing an electron transport chain and an enzyme called ATP synthase that we'll talk about more when we talk about chemiosmosis. So the electron transport chain, what happens is that electrons get passed through a series of these transmembrane proteins and carriers that are embedded in that inner membrane of the mitochondrion. So this picture here is if we were to take a mitochondrion like this and basically zoom into a section of the inner membrane here and look at it in this picture, this is what we would see. So kind of zooming in, we can see that this area here is the matrix. So this is that gooey kind of filled center of the mitochondrion. This area out here is the inner membrane space. So you would have the outer membrane of the mitochondrion kind of like out here. And then this is the inner membrane of the mitochondrion. And you might notice that it looks kind of similar to the cell membrane. And that's because it is very similar in its structure. So as you can see, it is a phospholipid bilayer similar to the cell membrane. So as you can see, we've got these kind of multiple protein carriers and channels that make up this electron transport chain. Now, we're not going to worry too much about the actual names of each of these different proteins. We really care more about what they do. And the main thing that happens is that electrons, as you can see, get passed through these electron kind of protein carriers. Now, where do the electrons come from? You've probably noticed that I'm using the color blue, and throughout the entire lesson, we've been talking about our NADH that I've been writing in blue, and we added that FADH2 when we talked about the citric acid cycle. Now remember, we put those little dots to remind ourselves that there were electrons there. So if we look down here, we've got our NADH and our FADH2. And what happens is that they give up those electrons that they picked up and they pass those electrons through these proteins. All right, you can see that for FADH2 or FADH2 as well, or also. So you'll notice that the NADH gets converted back into NAD+, and the FADH gets converted back to FAD. Now, so far throughout this whole process, we've been talking about how these are aerobic respiration steps, and we've talked about how aerobic respiration requires oxygen. But you may have noticed that we haven't talked about oxygen yet, and that's about to change. So oxygen really comes into play here. So you can see the oxygen here at the end of the electron transport chain. And what happens is that the electrons that come out of the electron transport chain actually bond to oxygen and some hydrogen in order to make water. So basically what happens is you have hydrogen and you have oxygen, and you pick up some electrons and you can make water in the process. So this is why you need oxygen. So this is specifically why you need oxygen in order for aerobic respiration to occur. Now, if you run out of oxygen, so if you stop breathing, what happens is that there's nothing to accept these electrons at the end of the electron transport chain. And if there's nothing to accept it, the electrons can't get passed through the electron transport chain anymore, because it basically gets clogged up with electrons, because those electrons have to go somewhere in the end. And that means that we don't recycle the NADH back into NAD+, and we don't recycle the FADH2 back into FAD. And these two are needed in order to be recycled so that the oxidation of pyruvate and, glycolysis, the oxidation of pyruvate, and the citric acid cycle, it needs these to occur. So if we are no longer recycling them, then it means that the cell no longer has the reactants it needs in order to keep going through aerobic respiration. So to summarize, oxygen specifically is needed at the end of the electron transport chain to accept the electrons, and we make some water in the process. And if we don't have oxygen, we can't accept those electrons, and it means that all of the previous reactions shut down. And if those previous reactions shut down, then we can't go through the oxidation of pyruvate, the citric acid cycle, and we can't go into the electron transport chain. All right, so we've talked about these electrons moving through the electron transport chain and ultimately getting picked up by oxygen at the end. But why do we care? The reason why we care has to do with these hydrogen ions here. All right, so these hydrogen ions, so this is just a hydrogen atom, and it's H+, which means that it's really just a proton. Remember that hydrogen is made up of one proton and one electron, but since it's an ion, since it's H+, it means that it got rid of its electron. It gave it away. So now, instead of being a proton and an electron, that electron is gone. So we're essentially just talking about protons getting transferred across this inner membrane of the mitochondria. So what happens is that as the electrons are getting moved through these proteins of the electron transport chain, they're providing energy in order to move hydrogen ions from the membrane, from the matrix, which is that inside gooey part of the mitochondrion, and moving it out to the inner membrane space that is between the inner and outer membrane. So the outer membrane would be like up here, and this is the outer membrane of the mitochondria. All right, and so what happens is that we ultimately end up setting up a concentration gradient for hydrogen. So we end up with a whole bunch of hydrogens hanging out in the inner membrane space, and not as many down here in the matrix, because all of the ones in the matrix are getting pumped out to the inner membrane space. So now that we've set up a concentration gradient for hydrogen, so we've got a lot of hydrogen ions in the inner membrane space and not as many in the matrix, we can then use that to basically make ATP. So this is kind of like water behind a dam. So as that hydrogen can move back through another protein, it can actually almost like turn a turbine in order to make ATP. So our kind of the last step of our aerobic respiration, remember that our fourth kind of overall step was oxidative phosphorylation. Oops. And then the first part of that was the electron transport chain. And now the second part of that is going to be our chemiosmosis. So what happens is that these hydrogen ions that are built up in this intermembrane space are going to start going down their concentration gradient. So they're going to go from high concentration to lower concentration. And they're going to do it through a specific protein, which is an enzyme called ATP synthase. And that's what this kind of enzyme is right here, kind of at the end of the electron transport chain. And if we look at the name of this enzyme, it tells us what it does. So ATP synthase. So first of all, ASE tells us that it's an enzyme. Synth should make you think of synthesis, which means to make. And then ATP, remember, is that energy molecule. So this protein is an enzyme that makes ATP. And so what happens is that these hydrogen ions then go down through this ATP synthase, and that includes all of these hydrogen ions that have built up here. They're all going through that ATP synthase. And as they do, that ATP synthase is taking ADP and phosphate and forming a bond between the second and third phosphates, the second phosphate of ADP and then that kind of free phosphate there. And we're ultimately making our ATP energy molecules. So to summarize oxidative phosphorylation, we have electrons that go through the electron transport chain, which ultimately combine with oxygen to form water. As the electrons are getting passed through the electron transport chain, hydrogen ions are getting pumped from the matrix of the mitochondria into the intermembrane space. And this sets up a concentration gradient for hydrogen ions. Those hydrogen ions then diffuse through the ATP synthase enzyme from their high concentration area to the low concentration area. And in the process, it turns that ATP synthase, so it like physically makes it turn in a circle, which basically transfers the energy from that turbine into forming a bond between the phosphate and ATP and a random phosphate that's just kind of hanging out in order to make our ATP. So let's take a look at how much ATP we made from cellular respiration. And remember that with cellular respiration, we're more specifically referring to aerobic respiration. So this was with oxygen, right? So we had oxygen involved, and this was going through those kind of those four stages of glycolysis, the oxidation of pyruvate, the citric acid cycle, and oxidative phosphorylation. So for each glucose molecule, so per glucose, here's what we get with regards to ATP production. So during glycolysis, we made 2 ATP. We actually made 4, but remember we had to invest 2, so we end up with a profit of 2 ATP. During the citric acid cycle, we made an additional 2 ATP. Now, the real kind of payoff actually happens during that oxidative phosphorylation where remember the NADH with the electrons and the FADH2 with the electrons. go through electron transport chain and chemiosmosis. Remember, during chemiosmosis, we had that enzyme called ATP synthase. So the end result from all of these reactions kind of put together, we end up with about 36 to 38 ATP molecules made per glucose molecule. So one glucose molecule, which is C6H12O6, provides us with a total of about 36 to 38 ATP. And that includes 4 ATP from the substrate level phosphorylation, and then about 32 to 34 ATP from our oxidative phosphorylation. So let's go over the kind of the summary of the reactions of aerobic respiration again, so that we can maybe more easily follow what's happening with the carbons, the ATP, and the NADH and FADH2. So first, let's start with glycolysis. So remember, in glycolysis, we start out with glucose, and glucose is a 6-carbon compound, and that glucose gets converted to pyruvate. Pyruvate is a 3-carbon compound, and we actually make 2 of those because of the glucose getting essentially chopped in half. Now that pyruvate then goes through the oxidation of pyruvate in order to form acetyl-CoA. And so the acetyl-CoA has coenzyme A coming in to form the acetyl-CoA. And we also have some carbon dioxide leaving. Now that acetyl-CoA goes into the citric acid cycle. And during the citric acid cycle, we do produce some carbon dioxide. Now remember that during these processes, we're also making some ATP. So during glycolysis, we actually have to put 2 ATP into the chemical reaction to get it started. And those 2 ATP get broken down into 2 ADP plus P. But we also make 4 ATP. So we have 4 of the ADP plus phosphate going in, and we end up with 4 ATP coming out. So altogether, we end up with plus 2 ATP during glycolysis. Now, during the oxidation of pyruvate, we do not make any ATP, but we do make some during the citric acid cycle. So we end up with 2 ADP plus phosphate going into the citric acid cycle, and we produce 2 ATP coming out. Now, in addition to basically rearranging or juggling these carbons and making ATP, we're also doing the redox reactions with our electron carriers. So during glycolysis, we have NADH go into the reactions where it picks up some electrons in order to form NADH. So NAD plus picks up electrons to form NADH. That also happens during the oxidation of pyruvate, where some NAD plus goes into the reactions where they pick up some electrons to form NADH. During the citric acid cycle, this happens with NAD plus, again, picking up electrons to form NADH. It also happens with another electron carrier called FAD, which picks up electrons to form FADH2. Now remember that the electrons picked up by NAD plus and FAD to form NADH and FADH2, these electrons go to the electron transport chain, So these electrons all go to the electron transport chain, which allows for oxidative phosphorylation to happen through chemiosmosis. So with these electrons going through the electron transport chain, we end up making an additional like roughly 32 to 34 ATP. So once again, the conversion of glucose to pyruvate is known as glycolysis. The conversion of pyruvate to acetyl-CoA is known as the oxidation of pyruvate. The citric acid cycle is what's happening here. And then the electron transport chain is the first part of oxidative phosphorylation. Which includes the events of the electron transport chain followed by chemiosmosis. So once again, glycolysis is first, followed by the oxidation of pyruvate, then the citric acid cycle, and then oxidative phosphorylation.